Good morning, everyone. Today I'm going to present on the topic of neurobiology of schizophrenia, and my moderator is Dr. Symptom ma'am. So first of all, localization of the symptom domain. So there are positive symptoms of schizophrenia, negative symptoms, cognitive symptoms, affective symptoms, and aggressive symptoms. The positive symptoms include hallucinations and delusions, disorganized speech, and that can be localized to the mesolimbic circuit. Then the negative symptoms, which are blunted affect, emotional withdrawal, poor rapport, and passivity, that can be localized to the mesocortical circuits and also to the nucleus accumbens. Then there are cognitive symptoms, which is difficulty with attention, problem solving, working memory, and speed of processing. Also, affective symptoms, which include depressed mood, anxious mood, guilt, irritability, and aggressive symptoms like verbal and physical abuse, self-injurious behavior, destructive behavior. These three symptoms, they can be localized to the frontocortical circuits. The neuroanatomical changes in schizophrenia. The neuroanatomical changes the whole brain. The most consistent observation is the macroscopic appearance of on the postmortem brain volume. There is small but significant reduction in the brain weight that can inversely correlate with the age of onset. The brain volumes are smaller in prodrome, time of first break psychosis, and in chronically ill patients. Studies have reported that reduced prefrontal and temporal lobe volumes in individuals with high genetic load or developing schizophrenia. Ventricles, there is enlargement of third and lateral ventricle. The enlarged ventricular volume is also found in unaffected relatives who share genetic material. Then there is prefrontal cortex. The postmortem finding in prefrontal cortex is volume deficit in the gray matter, that is 3 to 12% in schizophrenia. The volume decrease is not attributed to the neuronal loss, but rather to the reduction in the neurophils in the layer 3. So what is neurophil? Neurophil is an unmyelinated axon. Dendrites in the glial cell associated in the process is neurophil. And there is a reduction in the neurophils in the prefrontal cortex, in the layer 3 of the prefrontal cortex. Then there is hippocampal formation. So MRI studies find a consistent reduction in the hippocampal or amygdala hippocampal volume by 4% in schizophrenia that may be lateralized to the left side. The smaller hippocampal volume present at the time of the disease onset and in those with high genetic loading for the illness. The postmortem study suggests that an anterior-posterior gradient of the neuropathological finding, with the anterior part more affected than the posterior part. Smaller pyramidal neurons have been reported in the hippocampus. Then thalamus, in the mediodorsal nuclei, there is total neuronal count is decreased. These reduction in the neuron number are reported to be in the parvocellular and densocellular part of the mediodorsal nucleus, which project primarily to the DLPs. Basal ganglia. In basal ganglia, there is volumetric increase. Electron microscopic studies suggest that increase in the number of synapses in the caudate and the putamen, suggesting increased neuronal activity in the basal ganglia, and increase in the number of D2 receptors in the caudate, putamen, and also in the nucleus accumbens. The psychoarchitectural changes, that means changes in the cellular morphology, which are mainly seen in the prefrontal cortex, thalamus, and the medial temporal lobe. The volume changes are due to insufficient development of cell bodies and the neuronal processes of the schizophrenia nerve cells, which lead to reduction in the thickness of the cortex, decreased dendritic spines of the pyramidal cell neuronal processes, decreased length of dendrites in the cortex of the frontal and the temporal lobe, and decrease in the marker of synapses, synaptophysin, SNAP25, MAP2, syntaxin, complexin 1, 2, and GAP40. Now, the neurochemical changes in the schizophrenia. First of all, the dopamine pathways. So we have 5 dopamine pathways basically. First is mesolimbic pathway, which runs from the ventral tegmental area to the ventral striatum. Then there is mesocortical pathway, which runs from the ventral tegmental area to the frontal cortex. Then there is nigrostriatal pathway from the substantia nigra to the dorsal striatum, and the tubuloinfundibular pathway from hypothalamus into the pituitary gland. The dopamine hypothesis, which the classic dopamine receptor hypothesis, which is related to the dopamine hyperactivity in the brain. And this dopamine hyperactivity is due to increase in the sensitivity and the density of the D2 receptors. However, this hypothesis does not assume that dopamine hyperactivity fully explains all the symptom domains of schizophrenia. Then there was a modified dopamine hypothesis, which assumes that the schizophrenia is characterized by abnormally low prefrontal dopamine activity, causing negative symptoms that lead to excessive dopamine activity in the mesolimbic dopamine neurons, causing positive symptoms. Then the role of glutamate, that is NMDA receptor hypofunction hypothesis. So it is believed that NMDA receptor hypofunction occur due to genetic and other risk factors inducing epigenetic alteration leading to NMDA receptor hypofunction in schizophrenia. And this NMDA receptor hypofunction occurs at the GABA interneuron. So in this diagram, as we can see, this is a GABA interneuron. The main function of GABA interneuron is to regulate the functioning of the pyramidal cells in the cerebral cortex. So on the GABA interneurons, there are NMDA receptors. So in what happens normally is that when the glutamate, which is an excitatory neurotransmitter, is released from the pyramidal cell, it gives these glutamate will act on the NMDA receptor, which are present on the GABA interneuron, which will provide a negative feedback to these glutamate cells, pyramidal cells, negative feedback to these glutamate pyramidal cells. Now, if there is hypofunctioning of the NMDA receptors, this glutamate will not be acting on this NMDA receptors. So this negative feedback will not be given. So there will be hyperactivity of the glutamate, leading to overactive downstream glutamate signaling and dopamine release in the ventral tegmental area. Now, serotonin hyperfunction hypothesis, it assumes that 5-HT2A receptors on the glutamate neurons in the cerebral cortex are hyperfunctioning. This overactivation of 5-HT2A receptors may be due to an excess of serotonin or upregulation of 5-HT2A. And so if there is increase in the 5-HT2A serotonin, so there will lead to downstream increase in the glutamate and further release in dopamine in the ventral tegmental area. Then is GABA. Loss of GABA, there is loss of GABAergic neurons in the hippocampus. So GABAergic neurons, GABA are inhibitory neurons. So they could, because of which inhibitory neurons, they could lead to hyperactivity of the dopamine or dopaminergic neurons. So GABA is synthesized from the glutamate by glutamic acid D-carboxylase, GAD, which has two isoforms, GAD65 and GAD67. The reduction in GAD67 mRNA levels is found in the prefrontal cortex in schizophrenia. Renin, it is a protein secreted by GABA neurons, is involved in dendritic spine formation, is also decreased in prefrontal cortex and hippocampus in schizophrenia. Now, acetylcholine. So there is in the cholinergic neurotransmitter that is integral part of both cognition and memory function in schizophrenia, and it is disrupted. So there is decreased level of both nicotinic and muscarinic receptor. So abnormalities in the central nicotinic receptors and in the alpha 7 and alpha 4 beta 2 nicotinic receptors, and also there is a decrease in the M1 muscarinic receptors seen in DLPFC and hippocampus. Now, the genetics, schizophrenia, it is closely linked to the genetic factors. The heritability is 75%, and it is the second most common heritable psychiatric disorders. And this data can be derived from the population epidemiological studies, family studies, twin studies, adoption studies, linkage studies, and chromosomal aberrations and copy number variations. Now, population epidemiological studies, it says that the prevalence in the general of schizophrenia in general population is 1%. In non-twin sibling is 8%. If one parent is schizophrenic, then it is 12%. If both the parents have schizophrenia, then it is 40%. The family studies, this is basically pedigree analysis, and the data has shown that the family basis of schizophrenia with increased risk of developing schizophrenia in relatives of schizophrenic patients. In twin studies, the concordance rate in monozygotic twins is found to be 50%, and in zygotic twins it is 20%. Then the genome-wide association studies, they have identified more than 100 chromosome locus of candidate genes in schizophrenia. A few of the candidate genes are neuroglin-1, NRG1, which is involved in neuronal migration, connectivity, signaling, and myelination. So if there is a single nucleotide polymorphism in this gene, which will alter the mRNA transcript, which account for the role of NRG1 in schizophrenia. Now, DISC1, it participates in the hippocampal formation, and the its expression is high during neurodevelopmental period in the late fetal and early postnatal phases and continues into adulthood. Then there is this synaptobravin binding protein, DTNBP1, yeah, DIS binding. It is a key regulator of glutaminergic transmission and prefrontal function. Now, renin we have already studied. It is a protein secreted by GABA neurons involved in dendritic spine formation, and its expression is downregulated in blood and brain of subjects with schizophrenia. Then there is COMT, catechol O methyl transferase. It is a major dopamine catabolic enzyme. So, as we know that 22q11.2 chromosomal region is one of the well-established genetic risk factor for schizophrenia, and it increases the risk of schizophrenia development to 30 to 40%. So the COMT gene is located in this region and is in this microdeletion region. Now, there is D-amino acid oxidase activator, affects metabolism of NMDA receptor modulator D-serine. Other candidate genes are regulator of G protein signaling 4, proline dehydrogenase, and D-amino acid oxidase. Now, there is environment in schizophrenia, which says that the genetic factor present from birth may interact with the environmental factor across the life course to increase the risk of schizophrenia. So, the environmental factors can be obstetric complication, maternal infection, winter birth, childhood infections, migration, cannabis use, and urbanicity. So the neurodevelopmental model of schizophrenia, it states that the schizophrenia result from abnormalities in the development that begins in utero and continues through infancy, adolescence, and young adult. Two major periods of brain insult heighten the risk of schizophrenia: pre and perinatal insults and disturbance in a brain maturation during late adolescence. The pre and perinatal risk factors, the main three obstetric complications of pregnancy, that is bleeding, preeclampsia, diabetes, or RH incompatibility, abnormal fetal growth, development including low birth weight, congenital malformations in small head circumstances, and complications of delivery, asphyxia, uterine atony, emergency cesarean section. So these obstetric complications, they lead to ischemic damage, which lead to neuronal loss in the temporal brain regions. The genetic predisposition with environmental insult lead to altered excitatory inhibitory balance in the prefrontal cortex and reduce myelination that would alter the connective. Then is psychoneuroendocrinology. That is a stress vulnerability model, tonic phasic model of HPA axis dysregulation. So, what is vulnerability in psychosis? It is generally arise from a combination of early life genetic and environmental factors ranging from family history to obstetric perinatal complications to a consumption of cannabis. That is all the causes which are the vulnerability factors. Now, stress is a common mechanism by which the plethora of risk factors for psychosis confers their vulnerability. HPA axis mediate the influence of stress on the psychotic illnesses. So, if a patient is vulnerable, so the HPA axis will already be sensitized, causing overactivation of HPA axis as evidence from persistently raised basal cortisol levels in such patient, larger pituitary size, and a smaller hippocampal volume. So in healthy individuals, they have a normal daily cortisol level and retain a robust capacity for cortisol response to acute stress. But in vulnerable individuals who are also under chronic stress, this diurnal cortisol is elevated with associated reduction in the capacity for cortisol response to acute stress. So basically, in the vulnerable individual, the baseline cortisol will also be increased, but the capacity for the cortisol response to acute stress will be decreased. So this is a tonic phasic model of HPA axis dysregulation. Then there is neuroimmunology of schizophrenia. So immunologic abnormalities are associated, as we can see, the high heritability. Meta-analysis found that patients with schizophrenia exhibit signs of low-grade peripheral inflammation characterized by upregulated pro-inflammatory cytokines and acute phase proteins. A large genome-wide association studies has identified 7 significant loci in schizophrenia. The strongest association was found with the extended major histoincompatibility complex region of chromosome 6, MHC region on chromosome 6. It encodes for 200 genes that have a role in CNS and immune functions. So this region is also linked with the increased risk of autoimmune disorder. And this observation further strengthens the hypothesis that autoimmune disease can cause psychosis such as anti-NMD receptor antagonism. single nucleotide polymorphism in interferon gamma, which is involved in regulating both the innate and adaptive immunity, is associated with twofold increased risk of paranoid schizophrenia. Alterations in the immune system also influences the neurotransmission of neurotransmitters. So the immune system, they can activate indolamine 2,3 dioxygenase, which is an enzyme involved in tryptophan kynurenine metabolism. So kynurenine acid, it is an NMDA receptor antagonist in human brain. So increased level of kynurenine acid is found in the CSF of patients with schizophrenia. So this is the table showing the various inflammatory markers which are increased or decreased in schizophrenia. So the pro-inflammatory cytokines like interleukin-6, interferon gamma, tumor growth factor beta, and TNF alpha are increased. However, the anti-inflammatory cytokines like interleukin-10 and 4 are decreased. The acute phase proteins like C-reactive protein, haptoglobulin, alpha-1 antitrypsin, and alpha-2 microglobulin, they are increased. Antibodies against NMDA receptors and anti-cardiolipin IgG are increased. CD4 cells, CD6 cells, and CD56 cells are increased in patients with schizophrenia. Now, inflammation and schizophrenia. So, the activity of microglial cell is linked to the pathology of schizophrenia. So, what happens is that the various stressors like trauma and early life stress, maternal immune activations, pollution, toxins, the inflammatory cytokines that are increased in vulnerable patients, and the genetic risk factors all cause uncontrolled neuroinflammation in this patient. So a normal microglia or a unprimed microglia has the function of synapse maintenance and plasticity, synapse formation, and synapse pruning. So due to uncontrolled neuroinflammation, there will be due to uncontrolled neuroinflammation, there will be loss of this homeostatic microglia. So the altered microglia will have altered synaptic maintenance and plasticity, altered synapse formation, and pathological synaptic pruning and synaptic pruning. So there will be two hit, that is the perinatal insult or early life stress prime the microglia to act in hyper-responsive manner to later stresses and encourage shift to a pro-inflammatory M1 phenotype. So this is a two-hit hypothesis which says that two hits that early life stresses along with the genetic susceptibility of the individual cause inflammatory response of this microglia, which lead to loss of synapses due to pruning by microglia, lead to structural brain changes associated with schizophrenia and the development of negative and cognitive symptoms. Then the neuroimaging. So most of these morphological changes have been documented at the first episode of psychotic illness and may be progressive. So structural MRI and CT, we can see a reduced whole brain volume, reduced prefrontal and medial temporal gray matter volumes, then reduced thalamic volume, enlargement of the lateral ventricle and third ventricles, and diminished total white matter volume. Then a reduction in the hippocampal and amygdala hippocampal volume by 4%, lateralized to the left side. Basal ganglia, there will be volumetric increase in the caudate and putamen complex. There will be volumetric increase. Then in the functional neuroimaging fMRI studies, we can see a regional cerebral blood flow, which during rest, there will be no alteration. During active auditory hallucinations, the regional cerebral blood flow is increased in ventral striatum, medial temporal lobe, and superior and medial temporal gyrus. During working memory tasks, it is reduced frontal and DLPFC activation, and during problem solving or other executive functioning, there will be deficits in the DLPFC activation. In the PET scan, there will be increased glucose utilization in the temporal cortex during auditory hallucinations. Magnetic resonance spectroscopy showed prefrontal cortex deficits, increased membrane phospholipid degradation, and impaired synthesis have been reported in frontal lobes, and increased level of frontal lobe glutamine and glutamate. And the DTI, diffusion tensor imaging, white matter disorganization in the frontal and temporal subcortical zones are seen. These are my references.